So dear colleagues, good morning and welcome to this session on transporter-mediated drug-drug interactions translation into the clinics. That's organized by the Clinical and Translational section of IUPHA and the British Journal of Clinical Pharmacology. There's been some slight changes in the program, so we will have three speakers who will share their insights, research, and the practical experience in the field of transporter-mediated DDI, from in vivo assessments, biomarkers, to clinical perspectives. So our first speaker is Professor Martin Fromm. He's the director of the Institute of Experimental and Clinical Pharmacology and Toxicology and chair of Clinical Pharmacology and Clinical Toxicology at the Friedrich Alexander University, Erlangen, Germany. And the title of his presentation is Biomarkers for in vivo assessment of drug transporters. Martin?
So, dear Caroline, dear colleagues, thank you very much for the kind introduction and the invitation to this great meeting. I enjoyed it really a lot. And I would like to start with the following statements, and Kathy Giacomini nicely paved the way this morning already on these issues. So, as you have heard, during drug development, new molecular entities need to be tested as substrate and inhibitors of drug transporters. And as also Kathy pointed out this morning, in vitro extrapolations have still considerable limitations. So we end up with too many correct predictions, and that leads to a huge number of DDI studies which are conducted in the process of drug development, which turned out to be negative. And this is, of course, not quite ethical, because you expose healthy volunteers to compounds they wouldn't have to need if we would have better predictions models. And one approach is to use biomarkers, to use endogenous compounds for the prediction of transporter-mediated drug-drug interactions. And in order to reduce this false positive predictions, so in a way that in vitro data trigger these DDI studies in humans, and then they turn out to be negative. And we want, in the biomarker field, which has been quite active in the last couple of years, to reduce this false positive predictions. So, as I mentioned, there's a considerable interest in identifying endogenous compounds as potential biomarkers of transporter DDIs. And if you look, for example, at the draft version, the recent one of the ICH guideline M12 on drug interaction studies, they stated that the inhibition potential of a drug can be evaluated using mechanistic static models, PK modeling, or endogenous biomarkers. So before I come to a more precise example of one specific biomarker, I would like to go through some theoretical assumptions and discussions we and others contributed to on what criteria should fulfill an ideal biomarker for transporter DDIs. And there are, of course, issues of sensitivity, specificity, predictivity, robustness, and some other aspects which need to be considered too. So regarding sensitivity, of course, it's quite basic. The endogenous compound needs to be an in vitro substrate of the respective transporter of interest, which we would like to study. I put here the S after the transporter, because there will be some biomarkers which will be biomarkers for pairs of transporters, for example, some organic cation transporters located at the basal membrane and a respective transporter located at the apical membrane, and they both work in a coordinate fashion to promote excretion, and we might get some biomarkers for these two transporters together. And then the second question is regarding sensitivity. Do classical modulators or perpetrators of these transporters of interest really alter the kinetic endpoint of the endogenous compound. So is there a positive control? And of course, we would like to see dose-dependent effects. So do the increasing doses of the perpetrators also increase the kinetic endpoints of the respective biomarkers? Then we would like to see also sufficient sensitivity of the biomarker. So we would like to see that also weak or moderate modulators of these transporters have an effect on our biomarker. And finally, and Kathy nicely pointed out the example of these OATP1B1 polymorphisms, which affect, for example, statin disposition. We would like to see that a respective biomarker is affected by functionally relevant genetic polymorphisms of the transporters of interest. And this has been indeed shown for several OATP1B1 biomarkers I will mention later on. So regarding specificity, again, we need in vitro data, ideally showing that our biomarker is not a clinically relevant substrate of other transporters than the transporters of interest. And in vivo, we need negative control, so we need to show that prototypical inhibitors of other transporters, which are not actually in the focus of the respective biomarkers, will not affect the kinetics of our endogenous compound. Regarding predictivity, we have two issues. For example, first, we would like to have a qualitative prediction of an expected DDI. So will the biomarker react when we give a respective perpetrator drug, and will it qualitatively indicate, yes, we have to be careful? And second one, the more challenging one, is there also a quantitative correlation between the changes in the exposure of the biomarker and the changes of the victim drug in the respective DDI study? Regarding robustness, there's of course the issue that the biomarker should be stable and good to measure in our samples, plasma samples or urine samples. Ideally, it should have a low inter-individual variability, and this thing is not working all the time. Regarding inter-individual variability, we need to know factors changing biomarker disposition. For example, are there differences between ethnic groups, or are there gender-dependent concentrations of the respective biomarkers? Finally, regarding robustness, an issue which is of relevance for one of the biomarkers I will talk later on is there a circadian rhythm in the plasma concentrations or the urinary excretion of the biomarker? This is the case for some of the biomarkers and makes sampling and so on more difficult, and data interpretation too. We have to consider nutrition. So is it important to control food intake during our DDI studies, and how much is the biomarker changed in its plasma concentration to food intake? And then I think one very critical issue is better understanding the endogenous formation of the biomarker in the body, and also understanding the mechanisms of further metabolism of the biomarker in the body and its elimination. And this is, in my opinion, not very well studied for many of the currently proposed biomarkers. And yeah, that's related to what I just said. We have to consider carefully how the perpetrator drugs also might affect the formation of the endogenous compound in the body, and how the perpetrators might change the further metabolism of the endogenous biomarker in the body, because that, of course, will affect our measurements of the biomarker in the body. And then thorough studies need to be done to decide what is the ideal endpoint pharmacokinetic parameter to be studied. Is it AUC? Is it a single plasma concentration time point? Is it renal clearance? We have to decide on that issue. We have to decide on the minimum and the optimum sampling strategies, sampling time points and periods. Ideally, we would like to have the no further metabolism of the endogenous biomarker in the body, and would be nice to have autolog transporters and an autolog and a respective biomarker also in the animal model, so we can measure this concentration of the biomarkers already in animal studies before we go into humans. And we need to have an understanding on the impact of disease also on the biomarker disposition in case it will be used in patient populations. So you see there's a huge list of things we have to consider, and probably at the moment there's no ideal biomarker, and maybe there will never be an ideal biomarker, but we need to come as close as possible to these issues. So what is known, and there has been a really huge amount of literature coming up in the last 10 years on biomarkers, mainly for hepatic uptake transporters and for renal transporters. And you see here a hepatocyte with a number of uptake transporters located in the basolateral membrane and a number of transporters in the canicular membrane. And here you see certain biomarkers which have been proposed in literature. For example, here on the left side, the biomarkers for the OATP1B transporters, OATP1B1 and OATP1B3, which are the uptake transporters for statins, for example, which I mentioned before. And some of these well-studied biomarkers are the coproporphyrins, which show an association to this SLCO1B1 polymorphism, and which are also affected in their disposition when classical OATP1B inhibitors are administered. There are also some other bile acids and bile acid metabolites which are have been proposed, and you have heard in the talk of Kathy Giacomini this morning already the biomarker TDA and HDA. So there are a number of, and this field of OATP1B biomarkers is really quite well advanced and well studied. And there are other interesting biomarkers, for example, isobutyryl L-carnitine for OCT1. Here the uptake transporter for organic cations in the liver. And if we look at the kidney, you see here this polarized cell, again the basolateral membrane on the left side, the uptake transporters mediating uptake of cationic compounds or the anionic compounds, and then you have a variety of efflux transporters located at the luminal side. And if you look here at the cationic compounds, you have the uptake transporter OCT2, organic cation transporter 2, and it works in a coordinate fashion with the mate transporters. Classically drug substrate of these two transporters is metformin, for example, and as endogenous biomarkers have been proposed NMN and methylnicotinamide, N1 methyladenosine and creatinine, but the latter one has certain limitations because it has a really high fraction of filtration and a quite small component of glomerular of active secretion. And in the following, I will go a little bit more into detail on NMN as biomarker for this OCT2 mate axis. And the whole thing started, I think, largely with this very nice paper of the group of Professor Sugiyama and his colleagues. And they proposed N-methylnicotinamide as an endogenous probe for evaluation of drug-drug interaction involving the mates. And what is NMN? So it is a major metabolite of niacin, tryptophan, and nicotinamide. It has a high renal clearance. It has been reported to be between 260 and 400 milliliter per minute. So there's a pronounced active secretion of this compound. It's not protein bound in plasma. In vitro data show that it's a substrate of OCT2, MATE, and MET2K, MATE1 and MET2K. And also in vitro data indicate that classical inhibitors such as pyrimetamine and trimethoprim preferentially inhibit the MATE transport with higher potency compared to the OCT2 mediated transport of the respective substrates. So looking at a picture again, we have here this axis, OCT2 MATE, mediated polarized transport from the blood side to the urine. We have a number of drugs being excreted via this axis, and metformin I mentioned it as one example. And the hope is that the renal secretion of N-methylnicotinamide, for example, mimics in some way the excretion of the respective substrate drugs, and that inhibitors such as pyrimethamine affect the disposition of the clinical victim drug, metformin, for example, in a similar way as we affect the renal elimination of the biomarker, in this case N-methylnicotinamide. In this study by Professor Sugiyama and his colleagues in 2012, they nicely showed that the NMN renal clearance was considerably reduced when pyrimethamine as OCT2 MATE inhibitor was co-administered in comparison to healthy volunteers treated without this inhibitor. So this is a positive signal. This biomarker appears to be sensitive to pyrimethamine. We conducted a clinical study on the interaction of metformin and trimetoprene, and metformin renal clearance nicely decreased when this other OCT2 MATE inhibitor was co-administered. And in the same study, we found also a decrease of NMN renal clearance when trimetoprene was co-administered. So we have a sensitivity of the biomarker against two of the prototypical OCT2 MATE inhibitors, which is nice, and we have, at least based on this data, information on sensitivity of the biomarker. I took this figure from a very nice review of Dr. Rodriguez, which was published in 2022 in CPT. And you see here on the y-axis the decrease in metformin renal clearance due to a number of different OCT2 MATE inhibitors. And on the y-axis, from the same studies, the respective decrease in the renal clearance of NMN. And each black dot is one individual study. They use different OCT2 MATE inhibitors, mainly pyrimethamine and trimetoprene, and in different doses also. And there's really a very excellent correlation in these studies in the extent of decrease of renal clearance of metformin with the respective decrease in renal clearance of NMN. So that's very nice and comforting. Looking at the intra-individual level, the data become much more sparse at the moment. So there's, from our study, an analysis with a limited number of individuals. But if you look at, again, the change in metformin renal clearance due to the inhibitor, due to trimetoprene, and in the same individuals, you look at the change in the renal clearance of NMN, and there you find a reasonable correlation. We would like to see that also in larger sample sizes, and that's an issue we think we should also focus on in the future and get more data on this intra-individual correlation. So switching from sensitivity now to specificity, we used a collaboration with Boehringer Ingelheim. And as you know, Boehringer colleagues conducted cocktail studies where they try to identify drug transporter substrates, and they combined it in a cocktail and used respective perpetrator drugs and looking how the cocktail drugs react to the respective perpetrator drugs. But this is not the focus of our study. We had access to these samples and checked whether, first of all, the cocktail compounds affected NMN concentrations, which was not the case, but I don't show you the data here. And in a second step, we looked in the samples whether the NMN concentrations were affected by prototypical transporter inhibitors. So cimetidine as another OCT2 mate inhibitor was used in that study, rifampin as an inhibitor of OATP1B transporters, verapamil as a PGP inhibitor, and probenecid as an OAT inhibitor. And looking here at the results, on the y-axis you see the adjusted geometric mean ratio for the NMN renal clearance. And as expected, cimetidine caused in this study, again, a sensitive decrease in renal clearance of NMN. So again, sensitivity was proven. And the nice thing was that the other transporter inhibitors did not cause a significant change in renal clearance. We can conclude, at least based on this still limited number of inhibitors, that NMN renal clearance is at the moment sensitive and specific. There are, of course, limitations, as always, regarding NMN. One major limitation is this previously mentioned aspect of circadian rhythm. It is very pronounced for this compound. You see in the open curve with the open circles, the NMN plasma concentrations over time, over the day without an inhibitor, so the normal situation, and there is quite a pronounced increase from the morning to noon, and then there is a decrease throughout the day. And interestingly, OCT2 mate inhibitors like trimethoprim reduce this circadian variability. I'm not 100% sure about the mechanisms underlying both the circadian rhythm and this abolishment of the rhythm by OCT2 mate, but for sure we can say that we have to be really careful in the time points when we are sampling plasma samples and calculate renal clearance, or if you want to like to use just a single time point for predicting the DDI in plasma. So that's a challenging thing, and for sure we need to know for each of the discussed biomarkers, is there a circadian rhythm or not. Finally, I would like to share with you some unpublished data so far, again in collaboration with our colleagues from Boehringer. And in this study, we looked at 25 previously proposed OCT2 mate biomarkers, or also OAT biomarkers, which have been mentioned in the literature as putative biomarkers, or at least it has been mentioned that they are substrates of the respective transporters. Again, we used this study from Vivo et al., which I just described a minute ago, and looked whether the prototypical inhibitors, cimetidine, rifampin, verapamil, and probenecid, affected this biomarker amount excreted into urine in healthy volunteers. And the goal was to define first some decision trees so we could decide whether the respective biomarker is sensitive or not to the classical inhibitor. In the second step, we looked at specificity, and we were interested, of course, to see how many of these compounds were sensitive and specific to the respective, for the respective compounds. Here's the respective decision tree. I won't go through all the details, but in large, the first part was necessary to define whether the biomarker is sensitive or not sensitive. So we defined a certain threshold above which we considered the effect of the classical inhibitor as clinically relevant. So log4 2 change below -5 in response to the classical inhibitor cimetidine in this decision tree. And if the effect was also significant, meaning that the 95% confidence interval did not include 0, it was considered to be sensitive. And then in the second step, specificity was determined, and the same principle, so the effect of the other 3 inhibitors of verapamil, rifampin, and probenecid had to be small and not significant. And it turned out that among these 25 biomarkers, 6 out of the 25 were not considered to be sensitive at all. About 50% were sensitive but not specific, so the disposition of the biomarker was altered by the prototypical inhibitor cimetidine, but it was also altered by probenecid or verapamil, or maybe both of them. And in the end, it turned out that 6 out of the 25 were both sensitive and specific, using this respective decision trees. And of course, that 6th one would be the ones to look further in other studies and clarify them regarding in vitro data if they are missing and so on. These are not published yet, the data, and we hope to share further information on this issue in a due course. So taking together, I would like to conclude, we have certainly a considerable interest in reducing human DDI studies during drug development. There is a really large amount of literature around now, also coming from metabolomic studies on potential biomarkers. We have, as I mentioned, a relatively poor understanding for many of the biomarkers on their in vivo disposition, so mechanisms of formation and mechanisms of further elimination. We need also much more data on specificity of the biomarkers. It's not enough to show it's sensitive to a respective prototypical inhibitor of a transporter. No, we need to show it's also specific. And of course, we come to the issue which has been nicely discussed after Professor Giacomini's talk. Do we have specific inhibitors of the transporters? And there's, again, also quite a few things to do in the future. And in the end, we might end up with looking for one transporter family, OCT2 mate, not only at one respective biomarker, but a combination of several biomarkers, or we might combine biomarker approaches with cocktail approaches and in order to get best predictions for upcoming DDIs. Finally, I would like to thank my colleagues from my laboratory. I would like to thank the collaborators from Boehringer Ingelheim, and also I would like to thank the funding institutions, and I thank you for your attention.
Thank you very much, Martin, for the excellent presentation. The floor is now open for questions. Please state your name and country.
Great talk, Martin. This is Bhagwat Prasad from Washington State University. I was really impressed by the data on N-methyl nicotinamide and the promises, the drug-drug interaction that Dave showed, but then the challenge of inter-individual variability prediction. My question is more related to your lab has also shown nicely that in OCT2 mate combination, most of these drug-drug interaction could be driven by the mate mediated inhibition. And I was just wondering, these biomarkers fundamentally needs to have similar mechanism, right? The rate limiting step needs to be exactly similar to the drugs. So can you comment on that as a caveat, and how could we develop these so that we can have a specific mate versus rate limiting OCT2 biomarkers?
Yeah, thank you very much. Very good question. Yes, you mentioned, I was focusing on this biomarkers for OCT2 mate. So this biomarker reflects to some extent, should reflect to some extent the interaction for also drugs which are transported by the same transporters, and of course the affinity of the endogenous compound and also of the drugs might differ to the 2 of the transporters or 3 of the transporters, and this is an important thing. So at least for NMN, it has been shown that similar to metformin, the effect of pyrimetamine, trimethoprim is largely on the mate side in comparison to the OCT2 side. So we can roughly say, I would think that this interaction reflects more or less the same thing. But of course, it would be also nice to have a respective biomarker where the interaction is determined more by the apical side, the basal side, sorry. The DDI interaction dominated by the uptake and not the efflux. But I think NMN more reflects the dominated, mate-dominated renal excretion, and this is, in my notion, also the case from at least metformin.
Thank you.
Thank you. Any other questions? We've mentioned a lot about drug interaction with inhibitors. Do we have data on the opposite with inducers?
Biomarkers and inducers, I'm not so sure, because most of the studies are done with OATP and the OCT2 mate, and there's not too much of induction going on, and so I'm not aware of. So PGP would be a nice example, but there's, to my knowledge, no biomarker turned up at the moment. So I am not aware of induction studies with respect to biomarkers.
Thank you. So moving on to our next speaker, it's Professor Anne Nies. She's a head of membrane transport protein at Margarete Fischer-Bosch Institute of Clinical Pharmacology, Stuttgart, Germany. And the title of her presentation is Metabolomic Profiling and in silico ligand screening enable identification of novel drug transporter substrates.
Yeah, thank you very much for the kind introduction and also the kind invitation. And because we already had 2 great talks on transporters, I think I don't have to give a very long introduction, but I think a kind of a nice figure is that there are about 2,000 genes, 10% in the human genome are considered to be membrane transporters, channels, or have something to do with those 2 protein classes. So you already know that transporters contribute to the absorption, distribution, and elimination of various endogenous metabolites. We already heard that, and definitely also of drugs. There are currently 2 superfamilies that are most discussed, that are the solute carrier transporters. You already heard that in the introduction or in the keynote lecture by Professor Cassie Giacomini. And that are the ones that are discussed. Mainly the OATPs, OATs, OCTs, and mates. And there are also the ATP binding cassette proteins. There are only 48. And mainly there is the P-glycoprotein and the BCRP, as well as the multi-drug resistance proteins. So this is kind of another representation of the large SLC family. So there are there are currently 66 families, about 460 members. And what Professor Giacomini already said, there are many of those are understudied, and still about 30% without an assigned function. And that was the reason why we tried to develop a novel strategy and to identify SLC transporter substrates, and I will give you what we have done and describe proof of concept that is working to identify novel SLC transporter substrates. And we took 2 examples from the kidney. That is first the MATE1, we already heard about that, and the peptide-2. So what is MATE1? We just heard it. It's a multidrug and toxin extrusion 1 protein, and it's localized in the luminal membrane of proximal tubal epithelial cells, and there it exchanges protons with organic cations. So organic cations are effluxed into the urine, and protons are taken up. And it mediates the renal elimination of endogenous metabolites we just heard, such as creatinine or NMN, and also important drug is the metformin. The peptide-2, I think we haven't heard of that today. That's the peptide proton peptide cotransporter. It's encoded by the SLC15A2 gene, and it's also localized in the luminal membrane of proximal tubal epithelial cells. And this protein does a co-transport of small peptides and protons. So it's just rescuing the peptides which are excreted into the urine, so they are coming back into the body. And it mediates the tubular reabsorption of dipeptides and also peptide-like drugs like beta-lactam, antibiotics, and ACE inhibitors. So we set up a workflow, and I will go through that here in this slide. So we chose, in cooperation with 2 partners, knockout mice, either SLC47A1 knockout mice or SLC15A2 knockout mice. Then we prepared kidneys, not kidneys, but the kidney slices, homogenized them, and extracted polar metabolites. And also we collected urine. Then we subjected the kidney homogenate or the urine to hydrophilic interaction chromatography, in brief, HYLIC, and then did a Elsekutov MS analysis, and hopefully, or we expected to get something like that, a kind of a volcano plot where we can see endogenous metabolites higher or lower in the knockout mice. And then the hits we then validated in cell systems, either in double transfectants that express the OCT2 and MATE1, and they are grown polarized in those transfer membrane inserts Cassie Giacomini already introduced in the morning. Or we also generated HEK cells expressing peptide-2, and we can measure uptake of those compounds we identified into the cells. So cellular accumulation, and here we just looked what is flowing through. Then we hopefully identify compounds, endogenous metabolites, and then use the structures of those to do an in silico similarity screen using 2 drug databases that are the Drug Bank and Drug Central, and then come to drugs. And those we validated again in transporter overexpressing cells that are shown in number 4. Okay. So first, coming to the MATE1, what would we expect, or what did we see? So on the left side, you see a wild-type mouse. There we have the SLC22A2, the OCT2, and the MATE1, and an endogenous compound is the creatinine. And then you see that it's taken up into the kidney and then excreted into the urine. So what happens when you have a knockout mouse, that is shown on the right side? Then you don't have the MATE1 anymore, and the creatinine is accumulating in the kidney tissue. And, well, you can see that in the metabolomics analysis. You can indeed see that the knockout mice have much more creatinine than the wild-type mice. So that was pretty exciting, and we for sure looked in an unbiased fashion what is happening. You see here on the right side the volcano plot. So on the right side, going here, that is what is up in MATE, and we identified the creatinine, also methyladenosine and methylnicotinamide, and the propionyl carnitine, that were all compounds or substrates that had already been shown to be MATE1 substrates. So that was very exciting. And it was much more exciting to see those blue ones we identified, because that are novel substrates that hadn't been described before as MATE1 substrates. So particularly the methylcytidine, guanidinobutanoate, guanidinopropanoate, cytosine, and some acyl carnitines. And we then we selected those compounds from different classes and looked whether they are indeed transported in vitro. And as I said, we used that trans-well assay. So we added the compounds here to the basolateral side and looked what is coming out in the luminal side. And as a control, positive control, we used the metformin, and we did see nice transcellular transport. So the blank ones are always the control, so nothing is going through the control cells, but you need both the OCT2 and the MATE1 to get a nice accumulation of metformin in the apical membrane, apical compartment, sorry. And then we saw the same for those compounds, guanidino compounds, propanoate, butanoate, as well as acylcarnitine, cytosine, and methylcytidine. So we identified novel substrates of the MATE1. Not only in the mouse, but that are the human transporters, so that is for the humans. So then we wanted to look for another transporter, not just one, and we decided to look at the peptide-2. I already told you what that is doing, so it's recovering dipeptides from the urine, and here you would expect to have, in the knockout mice, to have novel compounds or compounds then down in the peptide-2 mice, and that is indeed the case. Here in the kidney, we identified one compound, that's the anthorine, that was down in the kidney of peptide-2 knockout mice. But then we also looked for urine in those mice, and now we are looking for something that is up in the urine of the mice, because that cannot be taken up again into the kidney. And here I don't know if you can see it, but all those, so we again found the anthorine, but we also found dipeptides here in orange as substrates that have, well, the dipeptides in general have already been identified as substrates of peptide-2, and apparently there was nothing else. At least we didn't identify anything else except for the dipeptides, so that was a nice confirmation of of the substrate spectrum. But because because anthorine hasn't been shown before as a substrate for peptide-2, only for the peptide-1, we also looked in the transfectants here, just an uptake intracellular accumulation, that the glycine sarcosine is a prototypic substrate, nice accumulation after 1 and 10 minutes. Again, that are the control vector transfectants, and that are the peptide-2 transfectants that are now the human transporter, and we could confirm the anthorine here. So then, we, as I already alluded to in the workflow overview, we then took the structures of those novel endogenous compounds and put those into the two databases, the drug bank and the drug central, and we identified or looked for cytosine and for methylcytidine, and identified flucytosine here in silico, as well as those compounds here, the cytarabine, salcitabine, telbibudine, gemcitabine, trifluoridine, capecitabine, and uridine triacetate, and with a score cutoff of 0.7. So we didn't identify any analogous substrates or substances analogous to the guanidino butanoate or the propionate or to anthorine. So that were the compounds or drugs we now had to test whether this prediction was okay. And indeed, so again, we used those double transfectants or even single transfectants in the transwell assay. So again, we put the compounds or the drugs down here and then looked what is coming out. And again, for almost all of them that had been predicted as novel drugs for the MATE1, we could identify compound transport into the luminal compartment. So the transcellular transport was confirmed for 7 of those, but not for this compound here. Okay, so we believe that's a combination of different research areas combining the metabolomics, cell biology, pharmacology, and data science. You can indeed identify clinically relevant, previously unknown drugs as novel transporter substrates. So that was the example for MATE1, nucleobases and nucleoside-derived anti-cancer and antiviral agents. But we believe that the strategy is not limited to the SLC transporters, but one can use it also for transport function of additional proteins such as ion channels or understudied transmembranes, the TMEM proteins, because the protein is, the transporter is always in its natural environment. And so that has a broad applicability for understanding translocation of compounds in general across membranes. Okay, with that, I would like to thank all colleagues from IKP that, who were involved in this study, as well as very good cooperation with Martin Fromm and Jörg König from Erlangen, and other cooperation partners from Germany, as well as from Japan, and funding agencies shown below. And I thank you for your attention.
Thank you very much for this great talk and promising results.
Thank you.
Are there any questions?
Great talk. Bhagwat Prasad from Washington State University. The metabolomics approach that you used is basically on the knockout mice model. I was just wondering more from the perspective of these knockout mice, if there are compensatory changes in the endogenous metabolome that could affect the signal. That's one. And then with respect to interspecies differences between mouse and human, for mate, for example, we know that the mate are expressed really highly abundant in mouse as compared to human. And then there is OCT1 also, which is exclusively present in mouse and not in human. So can that confound, although you have nice validation data, but your comments on basically the interspecies differences and the compensatory changes that could be happening.
Yeah, that are very good points, definitely. But we were very confident because we identified those substrates that had been described as MATE1 substrates. And we were, well, excited to find the novel ones, and that could also be then confirmed for the human transporters. But you are quite right that there could be differences, species dependent differences in those kind of experiments. But I think it's a good strategy to get maybe a completely new idea of substrates that are transported by transporters.
Thank you.
You're welcome.
Thank you. It's an excellent work. I'm Rao from University of Geneva, Switzerland. I rather have a naive question in relation to the metabolomic profile and then the metabolomic markers as a substrates for these transporters. We know that for xenobiotics, for example, alkalinization or acidification of the processes could sometimes changes the ionization, and then it will either eliminate or try to avoid elimination of the molecules. What are your thoughts on this acidification or the pH changes and pKa changes in relation to the metabolomic profiling in discovery as a substrates for transporters? To cut short, what are your thoughts on the role of pH in metabolomic profiling? You
mean on pH? Did I get it correctly?
Yeah, pH.
pH. Yeah, I mean, we looked, as I said, in the natural environment, kind of a natural environment. So the pH of the urine is, well, as it is, lower than what is in the cell. And I mean, for the in vitro experiments, we for sure adjusted the pH in the apical chamber for the MATE1 to 6.4, and in the cells it was 7.4. Okay. So we adjusted that in the.
So it's rather in the standard conditions.
Sorry?
It's rather under standard
yeah.
conditions.
Yeah.
But maybe in clinic, maybe that is something that that could change. Do you think that the changes in the pH in the environment could change the spectrum of the markers that we are going to find, or
well, that might be the case, but when the transporter is sitting in the, I think in the luminal membrane in the kidney, so it's exposed to that pH change, and so that it's the natural working environment, kind of.
Okay. Thank you.
You're welcome.
Thank you very much. Moving on from mouse models to the clinics, our last speaker is Professor Aleksi Tornio from the University of Turku, Finland. He's chief physician of the clinical pharmacology unit at Turku University Hospital. And he will give us his clinical view and investigation on transporters drug-drug interaction. Thank you.
Thank you, Caroline, for a nice introduction and invitation to give a talk here. It's very nice to be back here in Scotland. I had the pleasure of spending a postdoc here some years ago in Dundee, so it feels a bit like coming home here. So I will talk a bit more on the clinical side and clinical studies we have performed. I'm referring to here to the work that has performed at the University of Helsinki with Professors Janne Backman and Professor Mikko Niemi, with whom I have had the pleasure to work with and collaborate. But I don't have any unpublished data here. It's all been published already. So first I will go a bit to the clinical relevance of transporter-mediated DDIs in general, and I have few case examples how we can and have studied transporter-mediated DDIs. So about the clinical relevance, I start off with this slide or assumption that transporter DDIs and their clinical consequences are more complex than metabolism-mediated DDIs. There are several reasons for this, one of which is that you need to consider several sites during absorption, disposition, and elimination, and the localization of the transporters, and that they are expressed on multiple sites and with barrier function. And when comparing to just metabolism-dependent interactions where systemic concentrations increase, you can have differences in the tissue-specific concentration at the site of action, which might have impact on efficacy or adverse effects if the drug does not get inside the liver, for example. And as we have seen, there are usually multiple mechanisms involved, other transporters or metabolism, so it's not easy to pinpoint the exact mechanisms. Also in this sense, also in the sense what we have heard that there are not very good substrates and inhibitors that would be very selective. So about the clinical relevance also, if we look at clinically approved drugs, this is a few years back approved drugs by the FDA, we see that transporters come up there quite frequently as a victim of drug-drug interactions or as a perpetrator. What we see here and see in some further updates on this topic, that the transporters that really come up here are PGP, BCRP, and OATP1B1. there might be several reasons for this. One is, of course, that they are the most researched ones. We have good tools to study them, or best tools to study them, but also might be that they are the ones most relevant, but it is likely that we will see other clinically relevant transporter DDIs coming up in the future as well, once we learn more and have better tools to characterize them. And one example about the clinical relevance of transporter-mediated DDIs is about direct oral anticoagulants. Not so straightforward maybe, but at least factor 10 inhibitors like rivaroxaban are metabolized to a certain extent by CYP3A4 and 3A5, but not to that extent that they would be very susceptible to drug-drug interactions via that mechanism alone, but they are also substrates for PGP and BCRP and are excreted by them in the urine. This actually for rivaroxaban accounts for about 50% of its total elimination. And also, of course, if you consider PGP, BCRP, they are expressed in the intestine. So regarding drug-drug interactions, there's also the matter of increased bioavailability by PGP inhibition, for example. And what we see with these direct oral anticoagulants, we see increases in AUC by strong PGP CYP3A4 inhibitors of twofold or even more, which is kind of in line with the data presented there, considering the elimination and also the absorption by transporters. So drug transporters have a key role here in these drug-drug interactions, and also dabigatran is presented here, which is, or dabigatran etexylate is PGP substrate, where the mechanism is almost completely PGP inhibition in the intestine. So I would say this is a very relevant topic, but also shows that multiple mechanisms can be in play, and it's hard to disseminate which is the most important or what is the contribution. We are looking into this at the moment a bit, how we can see the contribution of the different mechanisms, but I don't have any data on that yet. But if we move on to how to study transporter drug-drug interactions clinically, of course we would like to perform clinical studies where we get an estimate of the pharmacokinetic effect and also understand the underlying mechanisms. And typically we would do this kind of pharmacokinetic study in healthy volunteers with an index substrate or index inhibitor, depending on the question we are asking. But for transporters, this might not always be possible, so we have several options which have dealt with a bit in the previous talks already. But I will go into that part next with some examples. So the challenge is really, as has been mentioned, to transporter DDIs is the lack of sensitive and selective index substrates and inhibitors, and also the complexity of the DDIs when we have several enzymes and transporters involved. So it might not be possible to study in a single trial thoroughly. And also modeling methods on their own are inadequate. And also it's not possible to study each situation clinically. So there are quite a lot of challenges. But how to then assess transporter DDIs and gain mechanistic understanding in clinical studies. I'm using OATP1B1 here as an example because it's one of the most well characterized. And there are, of course, two options here. We can evaluate a drug as a victim of an interaction or as a perpetrator in an interaction. So in the first question, we would typically use a selective index inhibitor, and the second question, selective index substrate, and work with that. But there are other options if we lack those inhibitors, and I will come to these options in the examples I have here. So regarding the probe substrates, there are several lists for these substrates and inhibitors, and some of the substrates and inhibitors are more selective and better than others. For example, for OATP1B1, for inhibitors, we have cyclosporine and rifampicin. Cyclosporine is maybe a kind of broader inhibitor of several transporters, mimicking more the worst case scenario, and rifampicin more selective towards OATP1B1, and it has been indeed well shown, for example, by this study by Mori et al. Rifampicin's impact on OATP1B1 substrate, and nicely demonstrating also the dose dependence of this interaction. And of course, when we talk about rifampicin, in this context, it's a single dose because we want to avoid the inducing effect here and have the transporter inhibition only in play. But if we lack this kind of selective or strong inhibitor, an alternative approach could come from pharmacogenetics. So we could do this kind of recruit by genotype study. A requirement for this, of course, is that we have this pharmacogenetic variability in the transporter that we want to study. That's a prerequisite for this. So we have individuals with genetically determined variability in their transport, their function. So instead of doing this kind of classic DDI crossover study with population not genotyped beforehand, we could approach this question differently if we are interested on the effect of a transporter on a substrate, or if a drug is a substrate for this transporter. So recruit by genotype individuals with different transporter function and dose the prospective substrate to these individuals and see if there are differences in pharmacokinetics. And going back to this classical study shown by Professor Giacomini, earlier by Professor Niemi, on the left-hand side, for simvastatin acid, this is clearly true that there is real difference between homozygous SLC01B1521 CC carriers versus TT carriers. Quite a big difference indeed, and in some of our newer work with a larger study with 170 healthy volunteers who were not genotyped beforehand, the same is visible here. So these poor function phenotypes are quite rare, but you clearly see a differentiation of the lower the predicted OATP activity based on genotype is, the higher the simvastatin acid AUC is. So kind of just substantiates this, that it's possible way, but of course you need this kind of variation that you can do this recruit by genotype study. But it could provide an alternative to this question. I will not omit completely when going into judging a drug as an inhibitor, and if we don't have an index substrate that is suitable, we could go with these indigenous substrates that has been discussed before. So I will just mention these glucodeoxycholate glucuronide and glucodeoxycholate glucuronide compounds that have been proven to be sensitive and specific OATP1B1 biomarkers. So showing here clearly as the top SNP the SLC1B1521, and also showing good differentiation, especially with the CC homozygotes with the control group, but also with the heterozygous showing higher concentrations compared to the control TT genotype. And this has actually been studied with an index inhibitor, same study as I showed before by Mori et al., studying different prospective OATP1B1 biomarkers. This seems to work quite well here as well. I have highlighted GCDCA glucuronide and the GGCA glucuronide here, so you can see here as well the dose dependence, which very nicely indeed. So as we gain more insight into these endogenous substrate, they could be they could complement or substitute these index substrates in some cases at least, but more work is needed. Then moving on to another part, how to assess different DDI mechanisms. An example with clopidogrel. The story starts over 10 years ago when it was reported in an epidemiological study, increased risk for rhabdomyolysis in serivastatin users with concomitant clopidogrel. And serivastatin that was already then withdrawn from the market is CYP2C8 and OATP1B1 substrate, CYP3A4 less important for its pharmacokinetics. The group did some in vitro studies, but the in vitro inhibition values were not low enough to substantiate a strong effect on serivastatin pharmacokinetics. But we got interested on this, and as we have focused quite a lot on CYP2C8, our favorite drug is repaglinide, which is CYP2C8 OATP1B1 substrate and CYP3A4 less important in pharmacokinetics, very similar to serivastatin, and did a clinical study with clopidogrel as a perpetrator with repaglinide, and it showed very clearly that clopidogrel increases repaglinide AUC fivefold with the loading dose, little less with the maintenance dose. And then we can have several mechanisms at play here, because there is CYP2C8, CYP3A4, OATP1B1 involved in repaglinide pharmacokinetics, and we know it is influenced by all these, its pharmacokinetics. And then if we look at clopidogrel and the data that we had on in vitro, we had indication on some inhibition on transporters and metabolizing enzymes. We did some further in vitro work at the time, showing in combination with the previous data that time-dependent inhibition of CYP2C8 was evident here, and one consideration here is the drug concentration. Clopidogrel, the parent drug systemic concentrations are very low actually. The glucuronide concentrations which was the SUB2C8 inhibitor, are several orders of magnitude higher. And based on this data, we built a PBPK model which indicated strong SUB2C8 inhibition and this kind of moderate OATP1B1 inhibition, about 50% of inhibition. So our next question, we were quite confident that yes, there is SUB2C8 inhibition here at play, but what is the clinical relevance of OATP1B1 inhibition by clopidogrel? And for that, we needed another clinical study to kind of know for sure. We could not rely on this modeling on its own. And for that, we used simvastatin. As we know, and I showed previously, simvastatin acid is a sensitive index substrate for OATP1B1, and also simvastatin lactone is a good substrate for or index for SUB3A4. And we saw no clear effect on either lactone or acid forms of simvastatin by clopidogrel. And this was similar results were gained by Professor Tsukiyama's group on pitavastatin. So it would seem that the effect on OATP1B1 is very limited. And we went on to do some other studies with SUB2C8 substrate showing clear changes in AUC of these SUB2C8 substrates. And the conclusion here really is that clopidogrel is not clinically relevant OATP1B1 or SUB3A4 inhibitor, but moderate to strong SUB2C8 inhibitor. Later on, we have done some work with the endogenous substrates for OATP1B1. I don't have the data here yet, but unsurprisingly, it seems that it does not have an impact on OATP1B1 endogenous substrates either. But this shows that we needed quite a lot of studies to conclude this and outrule at least any clinically relevant OATP1B1 inhibition by clopidogrel. And then lastly, I will move on to another transporter, BCRP and rosuvastatin as its substrate. Also substantiating here again the relevance of BCRP for rosuvastatin pharmacokinetics, ABCG2, 421 C2A SNV encoding BCRP has been linked with rosuvastatin pharmacokinetics. Again here, a previous study by Professor Niemi and some later work I have been taken part with Professor Niemi, where we looked at this on a larger scale with AUC from studies in healthy volunteers of a total of 247 healthy volunteers. The top SNP, or actually the only SNP coming up in the GWAS or the meta-analysis of the GWAS is the SLCA1B1, but in the candidate gene analysis we show an impact of ABCG2 similar to that we was shown by Professor Niemi's group before. So it seems that BCRP function has clear impact on rosuvastatin pharmacokinetics and in the clinical setting as well. So one study that we performed using rosuvastatin then as a BCRP index substrate was with febuxostat. So we dose pre-treatment with febuxostat for 3 days or placebo and single dose rosuvastatin, and febuxostat was beforehand suspected to be a BCRP inhibitor, and it turned out so. So rosuvastatin AUC was increased almost twofold by febuxostat, and also rosuvastatin transport was also evident in vitro study, kind of confirming this. But some caveats for rosuvastatin, as shown in some previous slides as well. Rosuvastatin pharmacokinetics is also influenced by variability in OATP1B1 activity, which could be due to genetic factors or due to non-inhibition of OATP1B1. So we have to be careful about the mechanism, what the mechanism actually is. And also, second point here, based on this, for these clinical DDI studies, it would be highly recommended to genotype the individuals for relevant pharmacogenetic variants to see if they have an impact on the results, and if there are these individuals with poor function of the transporters you are studying, you are not likely to see much of an effect on that transporter because it's already working poorly, so you cannot really inhibit anymore. Or if other transporters are working poorly that have an impact, then it can have an impact on your results. So it's good to see if pharmacogenetics is playing a role in the results. Of course, it's represent the population average altogether, but typically these are quite small studies, so it can have quite a lot of impact based on chance. But for the febuxostat-rosuvastatin interaction, it seems evident that the most likely mechanism is inhibition of intestinal BCRP. We have the in vitro evidence, and we saw this change in AUC and Cmax, but not on half-life or renal clearance of rosuvastatin. So these points substantiate the mechanism of inhibition of intestinal BCRP quite well. But this is kind of the key issue to design the studies in a way that you can gain an insight also on the mechanisms underlying the interactions, because it's only possible to perform a small number of drug-drug interaction studies, and to gain most information from them is the best way forward, of course. So really the take-home message or conclusion here is that designing and interpreting clinical transporter DDI studies really requires to account for the individual pharmacokinetic properties of the drugs that are being studied. There is no one-size-fits-all protocol for transporter DDI studies, and using carefully selected index substrates and inhibitors suited for the question you have, maybe complemented with endogenous biomarkers, will provide most insights into the mechanisms of the DDIs, but also multiple studies may be needed, and modeling is helpful, but not so useful without the clinical data, of course. And third point here is the pharmacogenetic variability. It should be taken into account always when we look at transporter mediated drug interactions, whether it has a role there, and in some cases, as mentioned previously, it can be used as a tool in this kind of recruit by genotype trials, which can may be used instead of these formal DDI crossover trials. And with this, I would like to conclude and thank Professor Niemi and Backman for these collaborations and this work, and I would like to thank the audience for your attention, and I'm ready to take any questions. Thank you.
Thank you very much, Aleksi, for the great talk and insightful clinical perspectives. Any questions?
Yeah, great talk, Aleksi. My question is more related to, I mean, are you surprised with the BCRP interaction that you saw at the intestinal level? Do we have any other evidence of BCRP mediated interaction affecting clearance? And my question is more because BCRP is mainly expressed in the intestine. The hepatic levels of BCRP is almost 100-fold lower or almost negligible as compared to intestinal level. And also in humans, unlike rodents, BCRP is not expressed in kidneys. So just wondering if there are other examples which show effect of BCRP on the clearance or THAF.
Yes, that's true, and I would say we are not surprised by this. So it's true. I don't know if there are any examples really affecting clearance as such, and also one point is of course that the concentrations of the inhibitor are higher in the level of intestine. So including that point here is additional thing that contributes to the fact that mostly relevance is the intestinal BCRP. Yes.
Yeah, thank you. Very nice data.
Hi, Richard Kim, University of Western Ontario, Canada. So interesting talk. Thanks for that. So with some of the statins, certainly rosuvastatin, multiple transporter involvement, and one of the things that is a little bit particularly difficult to interpret is the role of bile acid transporters. So we had shown that NTCP is a key transporter for rosuvastatin and other statins, but particularly in humans, and that diet, fasting versus food, can have an effect. So when you're designing these type of DDI studies, do you take into account role of NTCP or food?
Well, of course, it depends on the study. In this particular study that I referred to, the larger study include broad genotyping of these different transporters. So in that sense, took into account the effect of food is standardized in these studies. So it's a fasting state. We do see some effect on statins typically by food when they are not fasting for the whole day. So there will be some changes that are probably related to food intake later in the study and that, but of course we try to standardize the studies as much as possible, but of course the situation could be different if it was not a fasting state.
Great talk, thank you. So is there evidence for a sex-dependent effect on the transporter interactions?
For the interactions, I'm not certain if there are. For the interactions, we see clear differences. I think I have something here on the sex differences on the endogenous biomarkers, for example. So there are sex differences. How that translates to the DDIs, I'm not sure how much that has been studied, because typically these DDI studies do include male and female participants, or can do that, but the sample size is quite low, so it gets even smaller, so it can be a bit difficult to.
But generally speaking, is there sex differences in the function of the transporters? Significant difference.
Yeah, that's correct.
And are they consistent or are they variant? Like, are they generally females have more active or more, or they are dependent on the transporter itself?
Well, I don't have a definitive answer on my mind, but I would say that it's transporter dependent. We see some differences in other enzymes as well, so I don't have a general answer to this. I would say it's depends on the transporter, really.
Thank you.
Thank you for the talk. I have a question regarding assessment of drug-drug interaction on multiple dose versus single dose. In some of your examples, you showed Cmax after a single dose administration. Some of examples I can recollect it was day 1, day 3. So can you a little bit give your opinion? What is the best way to assess drug-drug interaction? Single dose, or after multiple dose?
Well, if we talk about the substrates, we typically would like to have a single dose of the substrate, and then try to extrapolate that to the steady state situation. If the pharmacokinetics is linear, that's quite easy, but for the inhibitor, it depends on the inhibitor and the questions you are asking. For rifampicin, it's clear it's single dose for OATP1B1, and the example I had was clopidogrel. The day 1 300 milligrams and day 3 75 milligrams was mimicking the clinical situation, where the 300 milligrams is the loading dose, which you start with in the clinical setting and then continue with the lower dose. So that was the reason for those two phases in that specific trial. But mostly for inhibitors, you would like to dose to get to the steady state, kind of to see what the worst case scenario is. But there are differences and different approaches in some settings, as mentioned.
Thank you.
Okay, thank you very much. Our sessions come to an end. I'd like to warmly thank our speakers for sharing their expertise and research in the field, and I thank you all for your attention.